Hello friends, I am Arpit Pandey and today we are going to discuss on a topic analytical method development for chromatography, basically for the HPLC. In my previous part I was discuss about the mobile phase selection, how we select the mobile phase, what is the logic behind it and what is the thumb rule we have to apply. And now today we are going to discuss the procedure for buffer selection. Buffer selection. So why we use buffer in a HPLC? Buffer are used in a HPLC mobile phase preparation in order to achieve reproducible chromatography. Buffers are needed when the sample contain ionizable species or if the sample matrix is acidic or basic in nature. In reverse phase chromatography, samples are sorry friends. In reverse phase chromatography, samples are separated based on their hydrophobicity. Okay, a compound in its ionized form becomes more polar and less retained in reverse phase liquid chromatography. Similarly, a compound in its unionized form becomes less polar and hence more retained in the reverse phase liquid chromatography. So remember this line: a compound in its ionized form becomes more polar. So ionized form it's become more polar and less retained in the reverse phase liquid chromatography, and similarly a compound in its unionized form becomes less polar and hence more retained in the reverse phase liquid chromatography. It means that more polar compound less retained and less polar compound more retained in reverse phase liquid chromatography. It is important to note that acid compounds are in ionized state as the pH increases, and basic compounds are in ionized state as the pH decreases. So with the help of this we can illustrate this thumb rule with the help of this graph. Means acid compounds are ionized in the state as the pH increases. Acid are ionized when the pH is increases, and basic are the ionized form when the pH is decreases. With the help of this graph you can easily remember this. So to develop a rugged method means a method in which the results are not varies as by changing the small but deliberate change in the matter conditions. Above, to develop a rugged method, buffer should be employed, preferably at least one pH unit away from the analyte's pK. This is derived from the Henderson-Hasselbalch equation. So you always listen that why pK we have to select the pH is the 2 units up or below the its pK value. This is because this equation is derived from the Henderson-Hasselbalch equation, where pH is equal to pK plus log A minus upon HA. Essentially, operating at a pH near the pK of the sample analyte means that it will be partially dissociated state. The analyte will be partially in its weak acid or base form and partially in its conjugate form. This will cause peak distortion in the chromatography due to poor peak reproducibility. Operating with a mobile phase at least one pH unit away from the analyte pK ensured that the sample would be in a single state almost entirely, that is either in ionized or unionized state. Once the desired pH range for the mobile phase is determined, choosing the right buffer can begin. Buffer capacity is optimized at or near a pH equal to a pK of a buffer. As a rule of thumb, most buffer works suitably well within the plus minus 1 pH unit of their pK. Here is the table for the buffer. You can easily remember. So phosphate buffer have three pKa values, that is the 2.1, 7.2 and 12.3. So if your phosphate buffer the phosphate buffer you selected and its pKa value is 2.1, so it is well worked within a range of 1.1 to 3.1 pH range, and its UV cutoff is 210 nanometer. Like the phosphate buffer, citrate buffer is also have a 3 pKa value, that is the 3.1, 4.7, 5.4. So previously I just stated that the pH buffer is worked well within the 1 pH unit below or upper their pK value. That is, if you choosing the citrate buffer and its pK is 3.1, so it is well worked within the range of 2.1 to 4.1 pH range. Format buffer, its pK is 3.8. Acetate buffer, its pK is 4.8. Tris hydromethyl ammonium methane, it's 8.3. Borate 9.2. Triethylamine 10.8. Another consideration for a buffer selection is the type of detector being used. For example, citrate may not be suitable for low UV applicants due to its high cutoff. If you choose a citrate buffer at a low UV cutoff, it will produce a so many noise and the ghost peak during the sample run. In some cases, buffer selection should be based on experimental observation and prior knowledge. Likewise, if mass spectroscopy detection is used, a volatile buffer such as TFA, acetate, and format should be employed. Use of buffer, ion pair reagent, and other modifiers such as triethylamine, diethylamine, TFA, etc. may be considered in the mobile phase to enhance the reproducibility, selectivity, or peak shape. When the use of simple buffer does not deliver desired result, the mobile phase with ion pair reagent can be attempted. Primarily, a buffer is used to regulate the pH and the acid-base equilibrium of the solute in the mobile phase. They may also be used to defer the retention time of the ionizable compound. The primary selected criteria of a buffer are buffer capacity and its effective range. First, second one is the miscibility of the buffer and its compatibility with the detector. Third, availability of the buffer to achieve the desired separation. Use of volatile buffer and modifier should be avoided for lower wavelength application. In general, the retention of analyte can be modified by changing the pH. The pH modification should be done by considering the counter ion part of the respective buffer, means because of the common ion effect. If you want to know about what is the common ion effect, go to my videos and watch the video on the topic common ion effect. The most commonly used buffer and pK value are summarized in table 3, where you can easily remember and know. In normal phase chromatography, in order to ensure the solubility of the analyte, the first step is to check the compatibility of the analyte with the solvent. Initially, what we have to do, one should start with N-hexane and N-heptane in the combination with ethanol or isopropyl alcohol. Selection of organic solvent in its ratio can be finalized during the optimization of the method. It is important to refer to the column manual before use and ensure the compatibility of the solvent with the column. All the chemicals and reagents should be of high purity grade, AR grade, spectroscopy grade, etc. Organic solvent should be of HPLC grade. Suitable modifier like trifluoroacetic acid, triethylamine should be used in the mobile phase if required. One should start with the minimum modifier concentration to prepare the mobile phase and progressively optimize the concentration. DA's mobile phase should always be used without filtration. So, how you select the mode of elution? For reverse phase, method development can be initiated by using isocratic or gradient elution based on the requirement. It is usually recommended to mix a small portion of an aqueous medium in organic portion to avoid the pumping problem. Similarly, a small amount of organic solvent in aqueous phase should be added to minimize microbial growth. Care should be taken to avoid the precipitation of a buffer in gradient mode of the mobile phase. It is also recommended to check the back pressure and physical observation and optimum pressure of the mobile phase. In gradient method, it is important to ensure that no analyte is eluted at the re-equilibration phase. It is important to provide for adequate re-equilibration with initial composition at the end of the gradient run. Also, it is important to ensure a consistent gradient pattern. And the next topic is the detector selection. How you select your detector during the method development. Here is the answer. Various detectors used in HPLC instrument include UV visual detector, photodiode, arrow detector, fluorescence detector, conductivity detector, refractive index detector, electrochemical detector, mass spectrometer detector, corona detector, or cat charge aerosol detector, evaporative light scattering detector, UV visual. In all of these, UV visual detectors are typically in many laboratories as they can detect a wide array of compounds. UV detector is preferred when chromophoric group are present in the compound and have adequate response for the detection. If any chromophoric group is present in your compound, then it is usually a UV visual compound. You can usually analyze with the help of UV detector. Analytical derivatization is mainly used for following reason. Sometimes most of compound are not UV visual, then we use the derivatization method to make it UV visual and analyze with the help of UV detector. Analytical derivatization is mainly used for the following reason: to permit analysis of compound with inadequate volatility and stability, and or to improve chromatographic behavior like peak symmetry, retention, resolution, and or to improve detectability. Example of functional group present in the molecule and suitable derivatizing agent are given here. If your compound is not UV visible and some derivatizing agent, some functional group are present in the analyte, that is amine, so you can easily go for the derivatizing agent, that is acyl chloride, aryl sulfonyl chloride, isocyanide, isothiocyanide, etc. Carboxylic acid, phenyl acyl bromide, methyl phthalimide, OP nitrobenzyl, N-diisopropyl linuria, NP-NBDI, etc. If your compound presents some hydroxy compound, so you can go for the derivatizing agent, that is acyl chloride, like benzoyl chloride derivative, phenyl isocyanide, etc. Carbonyl compound. If your analyte presents the carbonic compound, then you will use 2,4-DNPH, p-nitrobenzyl hydroxylamine, etc. Okay, so according to me, many always all maximum analyte presents the carbonyl group in it. So for 2,4-DNP and the acyl chloride is a very best derivating agent which is normally found in the laboratory, and easily all the derivatization is done mainly for the functional group which have amine, hydroxy compound, and carbonyl group. Derivatization can be done at the pre-column stage prior to the analysis, or at the post-column stage after chromatographic elution. The first detector is UV and PDA detector. PDA detectors are widely used to assure spectral peak homogeneity. It is mainly used during stress application for UV spectral identification and wavelength selection. Once you select a wavelength at which the highest response of the analyte impurity should be obtained. However, in some cases, a wavelength can be chosen different to the one for highest response in order to reduce the interference for the diluent, blank, or mobile phase, if any. Wavelength selection can also be done based on the optimum response of each component in the sample matrix. Dual or multi-wavelength strategies can also be adopted to monitor one or two components in a mixture if required. ELSD detector. The evaporative light scattering detector is far more useful for online lipid quantification than the commonly used UV detector. It works by measuring the light scattered from the solid solute particle remaining after nebulization and evaporation of the mobile phase. Corona detector, it is CAD. This type of detector is also very useful for not chromatographic compound. It operates by nebulizing the mobile phase from HPLC using a nitrogen gas stream. MALS detector, multi-angle static light scattering detector, perform absolute characterization of the molar mass and size of the macromolecule and nanoparticles in solution, offering superb sensitivity. The detector measures the intensity of the scattered light simultaneously at three angles. The molar mass value can be measured with the trios range from 200 dA to 10 mdA. The upper mass limit is primarily determined by the radius limit with the sensitivity at the level as 0.4 microgram per ml BSA and means 0.66.4 kDa macromolecule. The measurable RMS radius ranges from 10 to 15 nanometer and can be extended up to 150 nanometer with specific conformation models over a wide range of molecular weight and size and concentration. A mass detector should be used to determine molar masses, RMS radii, polydispersides, branching ratios, etc. Certain calibration constant and system parameter must be determined and enter in extra oversize. Otherwise, any other available detector may be used as per the applicability and analyte properties. Fourth one is the fluorescence detector. Fluorescence detectors are sensitive to the compound that are inherently fluorescent or that can be converted into fluorescent derivative. Refractive index detector. These are used in case in which chromophoric groups are absent in the compound. Since a refractive index detector cannot be used in gradient illusion, ELSD evaporative light scattering detector can be applied. So RI detector is you also call the universal detector, flow rate. How you select the flow rate of your method during the method development? These are used in cases in which chromophoric groups are absent in the compound. Since a refractive index detector cannot be used in a gradient illusion, flow rate of a mobile phase should be adjusted for desired retention, separation of analyte and back pressure and runtime. So it should be flow rate of your method should be selected during your practical runs. So column movement temperature, separation of analyte peak can be enhanced by selecting the right column temperature depending on the nature of the component. Higher column temperature reduces system back pressure by decreasing mobile phase viscosity, which in turn allow use of longer column with high separation efficiency. The overall separation and peak symmetry can be improved by simultaneous changes in column temperature and mobile phase composition. Okay, to read this, higher column temperature reduce symmetry back pressure by decreasing the mobile phase viscosity, which is in turn allow use of longer column with higher separation efficiency. Column oven temperature. Usually a column oven temperature within the range of 10 degree to 50 degree is preferred. If the column temperature is required to be more than 60 degree, then the capability of column packaging material and physical property of the same should be considered. Auto sampler temperature. How you decide the auto sample temperature? Auto sample temperature should be maintained between 4 degree to 25 degree based on the solution stability. If the solution is not stable at room temperature, then the auto sampler temperature should be kept at 10 degree or lower. One must be ensured that there is no recrystallization or precipitation of solutes when the solution is kept at a cold temperature. If the diluent is a fully organic solvent, there could be a possibility of evaporation of solvent from the vials. If these are not tightly crimped or repeatedly punctured, this could make the concentration of the solute different from the actual preparation and may adversely affect the result of assay. A cooled auto sampler is to be preferred in such cases. Injection volume. The injection volume of the chromatographic method should be adjusted based on the response of the analyte and the sensitivity of the instrument. A good starting point is that the injection volume should be in the range of 5 microliter to 20 microliter for HPLC. However, this can be varied based on the specific application or prior experience. In order to improve the peak symmetry and resolution, injection volume should be kept low. If the diluent composition is different from the mobile phase, it should be ensured that the injection volume of the method is within the calibration range of the system. If the desired injection volume of the method is beyond the calibration range of the injector should be calibrated for the desired injection volume. Sample preparation. For sensitive molecule, the material of construction and grade and quality of the volumetric flasks, pipettes, other measuring equipment and vials should be studied and defined in the test procedure. Material of construction should be glass, soda lime, borosilicate, etc., or plastic, polypropylene, polymethyl pentene, etc., and it should be transparent or low actinic. There are some important points to be considered while optimizing the sample preparation procedure. The first one is the sample matrix. It should be considered before optimizing the sample preparation procedure. First one is the sample matrix, second one is the sample concentration, third one is the diluent selection, fourth one is the extraction procedure, fifth one is the clarification procedure. It should be always considered. First is the sample matrix. These are some important points to be considered while deciding the sample matrices. Physical nature of the sample, it is solid or semi-solid or solution, suspension, etc. What is the physical nature of your sample? It is also very important. Drug to excipient ratio, stability of sample matrix, it is hygroscopicity, thermal, photostability, etc. Safety of API. Note: for low content and high potent drug, dropping of an intact tablet capsule is preferred. For high content drug, the tablet should be crushed or the content of the capsule should be collected and the required amount must be taken. Some coating material may interfere in the presence of grinding and hence the uniformity of the drug could be affected. Sample concentration. The concentration of the test and the standard should be set such that the response measured in 7 to 8 degree digit area. So, sometime I asked by many of you how much the sample concentrator is should be preferred. So, according to the me, the sample concentration of the test and standard should be set such that the response measured in 7 to 8 digit area for SA and RS may be achieved. Other concentration are also acceptable based on the chromatographic output. So, sample concentration of RS test should be optimized to achieve the LAQ below the reporting threshold. Sample concentration should be optimized in order to get the desired range in SA method. Diluent selection. Some important considerations are solubility, solution stability, solution immiscibility of the sample. This should be set preferably at twice the desired concentration, peak shape and resolution, aqueous organic ratio and pH of the diluent. Mobile phase compatibility. Note: mobile phase is the preferred diluent for the better chromatography. If the diluent is fully organic solvent, there should be a possibility of evaporation of the solvent from the vials. If these are not tightly creamed or repeatedly punctured, this could make the concentration of the solution different from the actual preparation and may adversely affect the result of assay. A cooled autosampler is to preferred in such cases. Extraction procedure. A typical sample extraction procedure includes sonication, homogenization, stirring, heating, cooling, vortex mixing, wrist action, mechanical shaker, partition, solid phase extraction, etc. Note: the sonication time and temperature should be optimized. The frequency of the sonication used should be noted. Some sonicator may not produce uniform frequency throughout its surface. Hence, it is important to study the sonication procedure by overcrowding the flask in the sonication bath or keeping the flask on various places of the bath. Requirements such as intermittent shaking, temperature, duration of extraction procedure should be studied thoroughly and defined in the test method. In the case of sample preparation by stirring, as such as stirring speed, time, temperature, and dimension of the magnetic bar should be defined. Multiple extraction procedure may be adopted for preparation of sample in order to obtain the desired accuracy. Multiple sample extraction procedures should be employed. For example, extraction by sonication procedure should be critically compared with stirring or with a wrist action mechanical shaker. Robustness study should be done for critical step of sample preparation procedure with different makes of equipment used for sample preparation. Sample clarification procedure. Sample solution for injection into a liquid chromatographic system should be free of undissolved particle. In a drug product, there could be insoluble excipients, and those should be removed by suitable means. Centrifugation, filtration, or the combination of both is the commonly employed technique to make sample solution suitable for LC injection. Occasionally, solid phase extraction and liquid partition are also used in sample clarification procedure. In case of centrifugation, it is important to optimize the RPM, that is the rotation per minute, and the time of centrifugation. If necessary, temperature during centrifugation should be studied and recommended. Syringe filter are widely used during clarification procedure. The ideal qualities of a filter are: no adsorption or degradation of the compound of interest. If you use the syringe filter, the ideal qualities for the syringe filter are: no absorption or degradation of the compound of interest, no leaching from the filters. This can be ensured by the absence of any extraneous peak in the chromatographic condition. Mechanical stability to withstand the filtration of high particulate suspension. Some of the commonly employed filter material and their characteristic given in the table number 6. That is, the first is the syringe filter we are normally found in the lab, that is nylon syringe filter, compatible with many HPLC solvent, that is THF, alcohol, acetonitrile, but not with strong acids. PVDF filters, polyvinyl difluoride, hydrophilic material for use with alcohol, hydrocarbon, biomolecules, ether, and acetonitrile. PTFE, polytetrafluorine ethylene, chemically resistant to almost all solvent, strong acid and bases. Hydrophobic membrane should be pre-wetted when used with aqueous solution. Polypropylene, hydrophilic material with resistant to the most acid based bases, DMF, DMSO, and alcohols, not recommended for use with hydrocarbon, esters, and solvents such as ACN. PS polysulfonate, polyether sulfonate variant, commonly used with the tissue culture and ion chromatography sample, is stable with many strong bases and alcohol, but few HPLC solvent, as it is hydrophilic, not compatible with the acetonitrile. CS cellulose acetate, used with aqueous solution and few hydrocarbons only, low protein in binding, hence good for many biological sample, not compatible with ACN or DMSO. Depending on the sample volume, the diameter of the filter should be chosen. Based on the application, the pore size should be chosen. The filter should be saturated with the diluent prior to the collection of the actual sample solution. For this purpose, the initial few ml of the filter solution should be discarded and the volume of the initial discarded should be defined. So, it is almost all about the method development for HPLC. In my next part, we will discuss about the force degradation study before finalizing the method. So, it is end now. According to me, its video become too long because I have to cover all the topics. Thank you very much for watching and listening all things. So, if you like the videos, so please share, subscribe my channel, and also share with your colleagues. Thank you very much for being with me. Thanks.